Hey Mustangs, in this video we're going to be taking a closer look at protein receptors and transduction pathways. So first, a quick refresher, a reminder of the three stages of cell communication. So we have the signaling molecule, also known sometimes as ligand, binding to a protein receptor. It could be on the surface or within the cell, so intracellular or a membrane protein receptor, and that is called reception. So that's when the signal actually gets received. Then you have the signal transduction pathway, and that's where you have a series of molecules that get slightly changed or modified and pass the signal along molecule after molecule after molecule till eventually you have a cell response where something gets activated or turned on, turned off, and it's going to change what the cell does for a short period of time. Now, one of the key things to remember as we go through this video is the idea of modification and change. How when a molecule or a protein, anything that you're talking about that's involved in this pathway, if it gets changed or modified somehow, it usually means it's getting activated. And when it becomes activated, it can change the molecule that comes after that. So it's a series of modifications and changes that activate molecules and pass on those changes one after another until finally something happens within the cell. So there's going to be three major types of protein receptors that we're going to take a look at here. So first we have G protein coupled receptors, Ligongated Ion Channels, and tyrosine kinases. So our first type of receptor is G protein coupled receptors. With a G protein coupled receptor, you have a receptor that is found in the cell membrane, and this receptor is going to have a spot for a ligand or a signal molecule to bind. Another part of a G protein coupled receptor is the G protein itself. That's where it gets the name from. Okay, so right here you have the G protein right here, and then you have usually another enzyme that gets activated as well. So first thing that's going to happen is a ligand or signal molecule will bind to that protein receptor that you find in the cell membrane. When it binds, it modifies that receptor, which basically that modification activates it. It's able to take the G protein, and the G protein gets replaced, the GDP with GTP. This is a molecule that's very similar to ATP, so there's energy there. So it gets modified, that G protein gets modified so that the GDP becomes GTP. And now we could say that the G protein has been activated. When it becomes GTP, it's slightly modified, slightly changes in some way, and now the G protein has been activated. That G protein goes to that enzyme that's also found usually in the cell membrane, and it's going to modify that enzyme, and then that enzyme could start a series of changes as well that lead to finally a cellular response. Okay. Now, G proteins, they are often used in embryonic development. They're used in sense of taste, sense of smell. There are many diseases, including these here, cholera, pertussis, botulism, where there's actually bacteria that create toxins, molecules that can interfere in the activation of a G protein coupled receptor. And G protein receptors are so important and involved in so many things that 60% of the medicines that have been created to this date actually involve in some way affecting a G protein coupled receptor pathway. So what we just saw here, many medicines are involved with that. So G protein coupled receptors are involved in many things, not just what we see here, but many, many things that go on within our bodies, within our cells. All right, next we have ligand-gated ion channels. And with the ligand-gated ion channels, notice this is also a cell membrane protein receptor. You're going to have a ligand that's going to bind to the receptor. It's gated. Part of the protein basically closes off, allowing any ions to flow through it. But when you bind the ligand, it causes the protein to be modified, so the receptor protein gets modified, it changes shape, and a result of its changing shape opens up the protein, allowing ions to flow into the cell, And when that happens, those ions can activate different types of cellular responses, so change different things that are going on in the cell. In order to close an ligand-gated ion channel, the ligand is simply released. That causes the protein to change back to its original shape, and the gate is not closed. So now the ions cannot flow through. Now, we're going to see this later on when we get into the nervous system, but this is a major way that signals are communicated through your body. Tyrosine kinase receptors, these ones are really interesting, and these have been found to be involved in many things within our body as well. So tyrosine kinase receptors, sometimes referred to as receptor tyrosine kinases. The name kinase, again, should ring a bell. The kinase means that it's going to be a molecule that's involved in phosphorylation, and in this case a protein involved in phosphorylation. So they have very unique shapes. And a large portion of the protein is found on the inside of the cell. You have the binding spot on the outside here. So what happens is a ligand or signal molecule binds to two different receptors. That causes those receptors to move together and form a dimer. Once the dimer's been formed, it's activated, but it's not fully activated to this point. So it's able to be considered activated. It's been modified. Remember, modification, it's not the same as it was here, so it's not the same as it was there. It's been modified, so the protein has been activated, but it hasn't been fully activated yet. And these tyrosines are going to be key. So it says TYR, TYR, and on 3 on each side here. Those tyrosines are actually simply amino acids, because remember, these are proteins, and proteins are made out of amino acids. So these specific tyrosines that are part of the protein here are a key part of starting a signal transduction pathway. So what happens is, here we see the dimer not fully activated, but activated. It takes 6 ATPs and it phosphorylates those tyrosines that are found on the dimer and the bottom portion of the protein here. Now we could say that this is a fully activated receptor, and it definitely has been changed. Remember, modification and change is a huge idea here, so it has been changed by adding these phosphates on. The next thing that can happen, other proteins that are involved in a cellular, a signal transduction pathway can bind to these tyrosines where the phosphates are located, and when they bind, that modifies those proteins, those enzymes could be, and it changes them, and they've become activated, and then they can go through a series of passing on changes to other molecules. The interesting thing about tyrosine kinase receptors is that each tyrosine can actually activate different molecules that lead to different cellular responses. So when you look at the other ones, it's usually one very specific cellular response that it's going to lead to. Here, you can actually do more than one cellular response when two different relay proteins, relay molecules, bind to two different parts and lead to two different cellular responses. Tyrosine kinase receptors are involved in metabolism in the cell cycle, so those are pretty big deals right there. And research has actually linked problems with tyrosine kinase receptors to cancers, diabetes, bone disorders, and even inflammation. So clearly these have a very, very important role in our cells and involved in communication and getting cells to do different things. All right. Last part of the video here, we have signal transduction pathway. The signal transduction pathway is basically a domino effect. So signal transduction pathway is the second step in cell communication. And during this step, you can kind of think of it as a domino effect, where originally the receptor gets modified and activated. That then can modify and activate another molecule. This one can then modify and activate the next molecule, and so on and so on. So you have a series of changes that get passed on. So the key here is the fact that one molecule, when it becomes activated, it can then modify and activate the next one, which modifies and activate the next one, and so on, until eventually you get to a change, an overall change within the cell. Now, two important things to remember about signal transduction pathways is that there are relay molecules. Relay molecules are almost always proteins. So there are proteins that are going to be modified to become activated, and then they can modify the next protein to activate it, and so on, and keep passing it on. Some of the most important ones that we're going to learn are protein kinases. There's that word kinase again. So protein kinases are proteins that phosphorylate. They're going to take a phosphate and add it to another molecule. And then there are secondary messengers. Secondary messengers are usually not proteins, and they're usually very small. And two really important ones that we'll see in the cell are cyclic AMP, sometimes modified as a lowercase c and then capital AMP, so CAMP. And then there's calcium. Calcium is another important secondary messenger. All right, so first example here, we have a phosphorylation cascade. Now, just by the name phosphorylation cascade, you should be getting the idea that this is probably going to involve kinases, so protein kinases. So relay molecules that are protein kinases that are going to phosphorylate other molecules. So here is an overall look at a pathway all the way from reception, transduction to response that involves a phosphorylation cascade. So here a growth factor acts as a ligand or signaling molecule. It binds to its receptor. The receptor becomes modified, passes on, and is able to become activated. It changes a protein kinase. That protein kinase, now that it's activated, can phosphorylate another protein kinase, and so on and so on. I'll show you on the next slide. You'll see exactly how this is happening. So you have a series of phosphorylations from one protein kinase to the next, till eventually a protein kinase can actually phosphorylate a transcription factor, which is simply a protein that's involved in protein synthesis, where you're going to turn genes on or off, and it phosphorylates that transcription factor here inside the nucleus. And as a result of that, a transcription factor, now that it's phosphorylated, becomes active, and now it can go ahead and turn on a gene, is what we're seeing in this case. So that's an example of a cellular response there. All right, so the phosphorylation cascade works in this way. So the growth factor binds to the protein receptor, modifying it and therefore activating it. Now that it's activated, it can modify and activate a protein kinase here. So the protein kinase, when it becomes activated, changes shape, is modified, so it changed. And now that it's modified, it's active. It's going to use ATP in order to phosphorylate the next protein kinase. So here's the next protein kinase here. Here's the first protein kinase, here's the ATP. They come together and are able to actually phosphorylate and therefore modify this, and you can see how it changed. Now that it's been modified, it is activated. It's going to be used to modify the next protein kinase. So in order to do that, it comes together with ATP, and it's able to modify that. So once it becomes phosphorylated by the other protein kinase, taking the ATP and breaking off a phosphate and putting it on here, it's become activated now. So you can imagine a series of these can happen over and over again, and that's how the signal transduction pathway works with the phosphorylation cascade. One protein kinase phosphorylates the next, which phosphorylates the next, and they're not activated until they become phosphorylated. All right, then we have secondary messengers. So secondary messengers, we're going to use a coupled G-protein coupled receptor here. So here's a G-protein coupled receptor. A ligand binds to it. In this case, we can use a hormone such as epinephrine. Epinephrine is also known as adrenaline. It binds to the G-protein coupled receptor. That then activates a G-protein. That G-protein activates an enzyme, and then that enzyme is going to activate cyclic AMP. And in order to do that, this enzyme here takes ATP and modifies it to become cyclic AMP. Okay? And that cyclic AMP is a secondary messenger. So now that it's been created, now that it's been modified and activated here, that can then activate something such as a protein kinase, which then continues a phosphorylation cascade maybe, which eventually will lead to some sort of cellular response, some sort of change within the cell, and it's usually temporary change. Okay? Now the big idea that you should get from these signal transduction pathways is that changes that happen to molecules during this pathway will wind up eventually leading to an overall change in the cell.